Do you ever wonder why learning biology is important? It's a springboard or jumping off point for many other sciences. Take the study of climate history, called paleoclimatology, for example. How do scientists who study paleoclimatology use biology to make correlations between ocean temperatures and the Earth's climate? Through biology. It starts on board a really special ship called the JOIDES Resolution that can drill into the seafloor and recover sediments and rocks from millions of years ago. On this expedition, scientists wanted to explore the bottom of the North Atlantic. The drill crew put a bit attached to a drill pipe through a hole or moon pool in the bottom of the ship in a process called tripping. When the ocean floor is reached, sometimes more than 2 kilometers under the surface of the water, drilling can begin. A 10-meter-long plastic core liner is fed into a metal core barrel and dropped down to just above the drill bit, and as the bit goes into the sediments and rocks, the core fills the core liner. Once it's pulled back up, the full core liner is pulled from the metal core barrel and it's brought to the core laboratory to be examined by the scientists on board. The cores are composed of a mixture of sediments and organic material that has drifted down through the water column over time. Because of this, the fossils of many organisms can be found in the sediment. The paleontologists on board the JOIDES Resolution can use them to help determine the approximate age of the sediment layers in the cores. When the core comes up, a small section of it is sent to the micropaleontology lab. Here, a scientist makes slides of the microscopic fossils, called nanofossils, which include coccolithophores. When alive, these unicellular, eukaryotic, photosynthetic algae float in the seawater. They have tiny skeletons made of calcium carbonate plates. The slides, containing pieces of the skeleton, called coccoliths, will be observed under the microscope to determine the species of organisms present, which will then help indicate the age of the sediment. Another paleontologist will process the sediment through a sieve that separates out organisms that are a little larger. These organisms are called foraminifera. Foraminifera are single-celled, heterotrophic protists. Forams can be viewed with a dissecting scope, so once separated from the sediment, they are put onto a tray for observation. These fossil shells are also made of calcium carbonate, like the coccolithophores, and like the clam and snail shells you find on the beach. The morphology of these organisms is known to evolve, so the species present and form of its shell can be used to determine the approximate age of the sediment. While the micropaleontologists are determining the age of the core for the other scientists on board, the cores are x-rayed and analyzed. The sections of core are then split down the middle into long halves so the scientists can see what's inside. Sometimes evidence of other types of organisms are present in the cores. Snail shells, urchin spines, and pieces of coral are a few of the fossils that can be found. Another type of fossil that is found are ancient disturbances, or bioturbations, of organisms as they bored through the mud on the ocean floor, and while the organisms are long gone, the evidence of them is preserved in the core. The scientists on board the JOIDES Resolution come from all over the world to work and study these cores. They work in the laboratories on board the ship in the middle of the ocean, making preliminary observations and measurements. This data will later be used by scientists to help determine the events that led to their deposition. Once all of the data is recorded for each of the cores, they are put in the ship's cold storage for the rest of the expedition. After the expedition, the cores are sent to their permanent home, called a core repository. These cores went to MAREM, a marine research facility in Bremen, Germany. After reviewing the preliminary data, each scientist decided which parts of the cores they wanted to study for their research. Once decided, they all got together in Germany to take samples from the cores. During this week-long sample party, the scientists got to discuss their upcoming research and collaborate on objectives while collecting almost 20,000 samples. These samples were sealed into labeled baggies and sent all over the world to their research facilities for further study. Dr. Dunkley Jones and Takuma Suzuki are two micropaleontologists that use biology to help solve the mysteries of the ancient climate. Dr. Dunkley Jones from the School of Earth Sciences at the University of Birmingham, UK, is using biomarkers from coccolithophores to check current climate models. Current climate models are based on the warming that occurred in the Pliocene and into the early Miocene, about 4.5 to 2.5 million years ago. The data that Dr. Dunkley Jones is gathering can be used to double-check these models and their predictions of the ocean temperatures in the North Atlantic. Coccolithophores produce alkenones, or fats, that change viscosity, or thickness, based on the temperature of the water. The colder the water, the more viscous the fats become. These properties can be measured in the lab and can indicate the temperature of the water during the time when the coccolithophores were alive. Takuma Suzuki from the Marine Corps Research Institute, Kochi University, Japan, is using the fossils of foraminifera found in the cores to determine how these organisms change morphology and the dependence of these changes on the temperature of the ocean water. His discoveries will impact the ability of other scientists to identify changes in water temperature through time. Takuma Suzuki is also analyzing the relative ages of these fossils to help in the ability of scientists to date sediments and rocks from the last 7 million years. Ocean currents in the North Atlantic are a part of the Earth's climate engine. Here, cold, salty water sinks to form a huge deep ocean current that circulates water and redistributes thermal energy on a global scale. The biological study of these microscopic organisms aids in the understanding of how the climate of the Earth has changed through time and what future changes may mean for the temperature of the Atlantic Ocean and important currents that begin there.